Over one-third of Americans spend an average of 5 hours a week enjoying the sun outdoors. This coincides with a high incidence rate of skin cancer in the U.S. 1 out of every 5 Americans develops skin cancer by the age of 70, with the risk of melanoma doubling for every 5 sunburns a person has. While these statistics clearly outline the prevalence of melanoma, the mechanisms currently available for diagnosis are painful, costly, and require a trip to the doctor.
To combat these issues, we've created a novel product, Solanoma. Solanoma is an at-home test kit for melanoma. It allows you to non-invasively test any spots on your skin that you think are infected by skin cancer. All you have to do is follow the simple instructions provided to you with the purchase of the kit.
First, you simply take a swab of the affected area on your skin. After transferring the cells within the testing area, you will follow the list of instructions in the pamphlet. This includes adding components A through G in order and waiting the proper incubation time between each solution. These solutions will be packaged in drop bottles for your convenience. After the final step, your results will be ready to view. This requires a spectrophotometer, which is available for purchase separately. Your results will be read accurately and quickly, giving you an answer to your question without even stepping foot inside a doctor's office.
The development of Solanoma consisted of vigorous cell work and analysis of fluorescence testing through aseptic techniques. A tyramide single amplification kit by Thermo Fisher was modified to isolate a protein that is found in vast quantities on the surface of melanocytes. This protein is called tyrosine. A biochemical reaction is required to interact with tyrosine. First, we must fix and permeabilize the cell. This means that we stabilize the cell while opening its membrane up. Next, we introduce a blocking buffer to prevent nonspecific binding. This way, we ensure that when we add solutions, we specifically target tyrosine and not other proteins. We consecutively add the primary and secondary antibodies. The primary antibody attaches to the cell membrane, while the secondary antibody attaches to the primary. Next, the secondary antibody connects to horseradish peroxidase. In this way, we've created a stable chain from the membrane to the peroxidase. Finally, our dye reacts with the peroxidase to activate the tyramide solution. This active dye will travel down to the cell membrane and attach to tyrosine, causing a fluorescent reaction. Through this process, we can detect the presence and concentration of melanocytes.
We modified multiple variables to find the most effective result this pathway could provide, such as cell density, secondary antibody incubation time, and tyramide reaction time. Over the course of the semester, we completed 29 experiments to find the best combination of these variables possible. We analyzed the result of each experiment through qualitative analysis using fluorescence microscopy and quantitative analysis using a spectrophotometer to read the absorbance levels. We also tested our procedure on fibroblast cells to ensure that Solanoma would exclusively identify melanocytes. After in-depth analysis of all our results, we created the procedure and final materials list that would be included in Solanoma. The kit consists of components A through G, multiple swabs, the testing container, and a pamphlet with the instructions.
Each kit provides 4 tests that can be used over a period of 6 months. Since the test is completely novel and the only non-invasive option on the market, it will cost $500 per kit. For individuals who spend extended time in the sun, physicians may recommend this product. In this situation, insurance may help cover the cost of the kit. An initial investment of $20 million is required to cover the first-year costs of renting a Biosafety 2 lab, renting equipment, paying scientists' salaries, and continuing research development. This will be recouped after selling 3,316 units. We believe this is achievable within the first year of sales.
While Solanoma is the final product of many hours of research, the product could be improved in the future in many ways. As the initial idea of this project was to produce a visual response by optimizing the biochemical pathway and including an optical component, future work could include focusing more on creating a light source that would produce a visual response when shown on the sample. This way, a spectrophotometer would not be necessary. Additionally, future work needs to be done on testing different types of cells and testing on samples taken directly from human skin. We hope that Solanoma helps you spend your time in the sun safely so that you can continue to explore nature for many years to come.
We would like to acknowledge Dr. Kishore and Dr. Basher for allowing us to use their resources to complete our experiments. We would also like to thank Dr. Nesnas and Dr. Ark-Remichev for their guidance as well. Finally, we would like to acknowledge our advisor, Dr. Keshav, for helping us through our many crises.